The last few topics we'll discuss in the biochemistry section are fatty acid and cholesterol metabolism. Do you remember from the very beginning of the metabolism section, where are fatty acid synthesis and degradation going to occur? Fatty acid synthesis is going to take place in the cytoplasm of a cell, whereas fatty acid degradation is going to mostly take place in the mitochondria of a cell. So let's go ahead and start with fatty acid synthesis, or how fatty acids are made. As you can see, our acetyl-CoA is going to be the substrate or the building block for fatty acid synthesis, but is also used as the starting substrate to make cholesterol and ketone bodies, which we'll cover shortly. The first step in fatty acid synthesis is going to be transporting this acetyl-CoA molecule from the mitochondria into the cytoplasm using our citrate shuttle, which in the process is also going to make a molecule of NADPH. So why is acetyl-CoA found in the mitochondrial matrix? Wouldn't it make more sense if it just started in the cytoplasm, which is where fatty acid synthesis occurs? Well, we have to remember a molecule of pyruvate is going to be converted into acetyl-CoA using our enzyme pyruvate dehydrogenase complex, which is going to be located in the mitochondrial matrix. This is why our acetyl starts off in the mitochondrial matrix and then has to make its way into the cytoplasm for fatty acid synthesis to occur. To think of this a different way, the acetyl-CoA must find a bus station in the mitochondria, hop on board the citrate shuttle, and get off at the cytosol shuttle stop, which is where fatty acid synthesis can finally occur. You'll see that fatty acid metabolism is going to take place in three primary locations. It mostly occurs in the liver, adipose tissues, and also lactating mammary glands. So once inside the cell cytoplasm, 2-carbon acetyl-CoA molecule is going to be carboxylated into a 3-carbon malonyl-CoA molecule. So this is going to be 2 carbons, this is going to be 3 carbons. We're adding a carbon group onto this acetyl-CoA. In the process, we're going to need the enzyme acetyl-CoA carboxylase. Like most carboxylation reactions, this vitamin requires biotin to be used as a cofactor. Now this step right here is the rate limiting step of fatty acid synthesis, and therefore it's going to be the most heavily regulated. Next, our enzyme fatty acid synthase is going to add 2 carbon units from malonyl-CoA to the growing fatty acid chain until it reaches a full length. This also requires NADPH, which conveniently we had made down here, and we can use that NADPH up here now. Subsequently, modification of palmitate can lead to further chain elongation, desaturation, or acylglycerol formation. Now that we've built up this very nice fatty acid molecule, let's tear it down. So let's go ahead and talk about the degradation side. Fatty acid degradation, also known as beta oxidation, a long fatty acid chain and CoA get converted into 2 carbon units of acyl-CoA by the enzyme fatty acid CoA synthetase. These acyl-CoA molecules are then transported to the mitochondria using the carnitine shuttle. The carnitine shuttle, which is inhibited by a buildup of malonyl-CoA, a substrate that we use to synthesize fatty acids. So notice that this is going to inhibit the carnitine shuttle. Why would malonyl-CoA inhibit our carnitine shuttle? Well, that way you don't break down fatty acids when you're actually trying to make them. So once we've successfully moved our fatty acyl-CoA into the mitochondria via our carnitine shuttle, we encounter the enzyme acyl-CoA dehydrogenase, which makes a double bond in acyl-CoA, which is required for our beta oxidation to occur. This results in acetyl-CoA molecules, which can then enter our TCA cycle or be used directly for energy by making them into ketone bodies. If all else fails on test day, just remember that citrate shuttle is involved in fatty acid synthesis. The carnitine shuttle is involved in the carnage of fatty acids. When everything here works correctly, our fatty acyl-CoA is going to use the carnitine shuttle to enter our mitochondria. However, what would happen if this carnitine shuttle wasn't working correctly? In patients with systemic primary carnitine deficiency, there's going to be an inherited defect in our carnitine shuttle, which prevents the long-chain fatty acids from entering the mitochondria. So it's going to inhibit our carnitine shuttle from working properly. These patients will usually present with generalized weakness, hypotonia, and a hypoketotic hypoglycemia. So why would it be hypoketotic? Well, during times of hypoglycemia or low blood sugar, our body turns to fat for reserves for energy. If we don't have our carnitine shuttle working properly, we get a toxic buildup of fatty acyl-CoA that's then stuck in the cytoplasm of a cell. This fatty acyl-CoA cannot get converted to ketone bodies or into our TCA cycle to make energy. So in a patient with systemic primary carnitine deficiency, you will see a hypoketotic hypoglycemia. What do you suspect an acyl-CoA dehydrogenase enzyme deficiency will cause? Well, if you don't have the acyl-CoA dehydrogenase enzyme, beta oxidation cannot occur, so fatty acids cannot get converted into acetyl-CoA molecules. Without acetyl-CoA, glucose and ketones cannot be formed, and this ultimately results in an increase in dicarboxylic acids. Medium-chain acyl-CoA dehydrogenase deficiency is an autosomal recessive disorder which leads to the buildup of 8 to 10 carbon fatty acyl groups building up in the blood. Again, the clinical presentation will be a younger patient who may have this hypoketotic hypoglycemia, the same that we saw earlier. This patient may also have lethargy, seizures, vomiting, or liver dysfunction. How are we going to treat a patient with this medium-chain acyl-CoA dehydrogenase enzyme deficiency? Well, let's think about it quick. When do we need to break down our fatty acids? When do we need beta oxidation? Well, we use beta oxidation to break down fats when our blood glucose levels are going to be low. So treatment is going to be keep our body supplied with a constant source of glucose and avoid fasting states. From our discussion of fatty acid metabolism, we talked about how fatty acids can be converted into acetyl-CoA molecules, which can then enter our TCA cycle. However, these acetyl-CoA molecules can also be turned into ketone bodies. So let's go ahead and talk about our ketone bodies now. The ketogenesis pathway is shown here. I'm not going to spend much time on this pathway since it's not very high yield for boards. I'll touch upon what you need to know. What are the three main ketone bodies? The ketone bodies will include our acetoacetate, our acetone, and our beta hydroxybutyrate. So what's the point of making these ketone bodies? When the amount of glucose available is not sufficient for the energy needs of the brain, so in other words, a hypoglycemic state, free fatty acids are going to be used as a rich source of energy. The liver can convert free fatty acids and ketogenic amino acids into ketone bodies, primarily acetoacetate and beta hydroxybutyrate, which can then be metabolized by the brain and the muscle into two molecules of acetyl-CoA for energy. Since ketones can be used in the mitochondria of the cell only, they cannot be used by erythrocytes, since erythrocytes or red blood cells have no mitochondria. In a prolonged starvation state and diabetic ketoacidosis, oxaloacetate must be used for gluconeogenesis, and therefore this oxaloacetate becomes depleted from our TCA cycle. Also in alcoholism, excess NADH is going to convert oxaloacetate to malate, which we discussed earlier. Both of these are going to cause a buildup of acetyl-CoA molecules, which now get converted into ketone body formation. Why are ketone bodies important clinically? Well, acetone, one of our ketone bodies, can give one's breath a fruity odor, which can indicate that somebody is in a diabetic ketoacidotic state. So, diabetic ketoacidosis can be partially determined by one's breath on whether acetone is present in one's breath, the fruity breath smell. One final point is that the liver makes most of these ketone bodies, but it lacks the enzymes to use them. So it'll actually make these ketone bodies, but the liver itself is not going to use these ketone bodies. There's a urine test for ketones, but it really doesn't detect the ketone beta hydroxybutyrate. We should do a clinical example using ketone bodies. An 18-year-old type 1 diabetic stops taking his insulin and eventually develops a fruity smelling breath and ketone bodies in his urine. Why is this? This man developed ketoacidosis because he depleted his oxaloacetate since his body ramped up gluconeogenesis formation. Since he depleted his oxaloacetate, his TCA cycle then stalls, which is going to shunt glucose and free fatty acids into ketone production to supply fuel for both the brain and the heart. Up to this point, we've talked about many different types of fuel use, including glucose, fatty acids, and ketone bodies all as fuel. Hopefully you'll now be ready for any pathway related questions, but you may also see a question on step 1 which asks which fuel source is used in each scenario, such as during exercise, the fasted state, the fed state, or in starvation. Let's go ahead and create a graph or a figure that can give you an idea of which fuel source is used during exercise and how long each fuel source can last. In the very first few seconds of exercise, we're just starting off our marathon. We're going to be using stores of energy, such as stored ATP, creatine phosphate, and then to a lesser extent, anaerobic glycolysis. These are all going to provide our energy in the first few seconds. After those first few seconds, the stores of ATP become depleted, and thus anaerobic metabolism, seen here as the green line, is going to start to ramp up. Once the body realizes that this exercise is going to continue, notice how we're running a marathon, the body is going to ramp up its aerobic metabolism, seen here as the yellow line. When exercising for hours, remember we're running a marathon here, both glycogenolysis, which is the breakdown of glycogen, and free fatty acid oxidation are going to be used to make ATP during our aerobic metabolism. So both glycogen and fat is going to be used. Here we're going to see the overall performance line drawn in. Notice how as time goes on, the body is going to have to rely on different fuel sources for energy, and as a result, the overall performance one can accomplish goes down, which is why you don't have as much energy at the end of a marathon as you do at the beginning. And as a side note, some stored glucose is going to be conserved by the body until you really need it. We're going to deplete most of our stores of energy, but some glucose is going to be reserved by the body for when times that you need it, such as sprinting at the very end of a marathon, sprinting to the finish line. Your body has some reserves of energy left. So that was our scenario during exercise. Now, how does the body react during fasting or starvation? In these scenarios, the priorities are to supply enough glucose to the brain and red blood cells so that the body can maintain the basic functions, and also to preserve protein to prevent muscle breakdown. Let's go ahead and create a scenario. You decide to binge on fast food cheeseburgers. After the third double cheeseburger with large fries, you step on the scale and realize you've gained 350 pounds. Now you decide that you're not going to eat for 5 days to lose some weight. In the fed state, immediately after eating those burgers, insulin is going to be used to stimulate the storage of lipids, proteins, and glucose into cells. Assuming that the body has plenty of oxygen available, both glycolysis and aerobic respiration can occur. After a few hours, the body will enter a temporary fasting period. Between meals, our body is going to start producing the hormone glucagon. So what does glucagon do? Glucagon is going to cause the body to use fuel reserves to maintain bodily functions. Hepatic glycogenolysis is the main way the body refuels after fasting between meals. This is going to be supplemented by gluconeogenesis and fatty acid metabolism. Unfortunately, glycogen stores are only going to last about 1 day. Remember that we ate those 3 large cheeseburgers and we will now not eat for 5 straight days. Initially, our hepatic glycogenolysis would have supplied the body with glucose. After the first day of fasting, you now enter the starvation phase, in which case you must use other fuel sources to make ATP. Like we saw before, glucose levels are maintained by hepatic glycogenolysis. After most of the hepatic glycogen has been consumed, the body must get creative now to find alternative sources for energy. Adipose tissue comes into play. Adipose tissues are going to start releasing free fatty acids for beta oxidation into the bloodstream. This means that muscle and liver must switch their fuel source from glucose to now fatty acids. The liver can use these fatty acids, as well as lactate and alanine, for gluconeogenesis to supply the body with more glucose, which is especially useful for red blood cells, since they cannot use fatty acids or ketones themselves. So that leads to the question, why cannot red blood cells use fatty acids or ketones? Well, it's because they lack mitochondria, so they need glucose instead. After day 3 of starvation, ketone bodies become the main source of energy for the brain and the heart. During this phase, about 2/3 of the energy used is from ketones, whereas about 1/3 is still from glucose. Since these ketone bodies are made from fatty acids, the amount of stored adipose tissue determines the survival time, assuming that you have access to water and other important vitamins and minerals. Once these adipose tissues are depleted, vital proteins are then degraded to make energy, which doesn't take long to result in organ failure and death. So what's the moral of the story? Do not binge on fast food because starvation makes your body scramble for different sources of energy. In the last unit of our biochemistry metabolism section, we're going to talk about cholesterol metabolism, lipid transport, lipoproteins, and dyslipidemias. Cholesterol is an important compound of cell membranes and is also the precursor for bile acids, steroid hormones, and also vitamin D. It can be synthesized in the smooth endoplasmic reticulum of any cell in the body, but most of it's going to be made by the liver. Once cholesterol has been synthesized, about 2/3 of it is then esterified by the enzyme lecithin cholesterol acyltransferase, or LCAT for short. What is the point of this esterification by the enzyme LCAT? Well, the esterification of cholesterol allows it to be stored inside vesicles rather than embedded in the cell membrane, which is important so that it can be transported through the blood to other parts of the body. Here's this big, complicated, and truthfully useless chart for cholesterol synthesis. You may be thinking, do I need to know all of this? The answer is no. So let's go ahead and focus on the important parts. Acetyl-CoA is used to make HMG-CoA, which can then be used to synthesize either ketone bodies or, in this case, cholesterol. The important enzyme HMG-CoA reductase, which converts the HMG-CoA into mevalonate, is going to be the rate-limiting step in cholesterol synthesis, and therefore is a good target to regulate our cholesterol production. Statin drugs, statin being the suffix. So statin drugs such as atorvastatin are going to competitively and reversibly inhibit the rate-limiting enzyme HMG-CoA reductase. If you inhibit this enzyme, you decrease the cholesterol levels by decreasing the cholesterol synthesis pathway. The boards love this enzyme, so make sure you commit it to memory.